people begin to join us, I'll just kind of introduce you all to our second Applied Catalysis Group and UK Catalysis Hub joint meeting. So the theme of this meeting today is on applied characterization. And characterization is something that's been certainly a feature of my research career all the way through it, from PhD, when I used things like 2-photon spectroscopy and lasers to try and look at reactions, to now where I'm looking at artificial metalloenzymes and XFs and trying to deal with things in water. And I'm sure we'll see a lot more today about this area and looking at catalysts and how we can characterize them both in situ and in depth. So we've got 4 great speakers today, both from academia and industry. And so to start off, we have Janelle Steves from GSK. So she's joined us all the way from the USA. She's part of the GSK Chemical Development Group, and over the last few years has developed processes for the pilot plants and expanding the high-throughput experimentation platform. So I'd like to pass over to Janelle and welcome her today and start her talk on HTE in 3D. Over to you.
Okay, thanks Amanda. Let me try to share my screen here. And if somebody could confirm that they can see, that would be great. Okay, so can you confirm that you can see this?
Yes, we can see
Great.
that.
So thanks for the invitation to present today, and I'd like to thank Catherine Wheelhouse for introducing me to this group. Catherine has been my colleague in Stevenage. We're both very experienced in the realm of catalysis, and we've talked a lot over my past 4 years since I started at GSK. So I guess I'll start by saying I kind of am going to present a different take on the term applied characterization and talk about how we can characterize molecules computationally to design better high throughput experiments. And I will apologize to the whole audience right up front, because I only realized last night that since daylight savings started in the US this past weekend, we're now in this weird 2-week time frame where we just have a 4-hour difference between Eastern Standard Time in the US and Greenwich time in the UK. So I do have a training course that I need to pop out for right after this. So I'll take probably 1 question right after I'm done with my talk, if we have time, but you can feel free to email me or connect with me on LinkedIn if you have other questions. And Catherine and Corinne obviously have my contact information. So as you can see, the title of my talk is HTE in 3D, and basically all of the work I'm going to show you was done in collaboration with my computational chemistry colleague at GSK, Jen Elward, who is also in the US, and we've worked together over the past 4 years in this area that we're going to talk about. So I was kind of interested during a rotation that I had in the computational chemistry group with Jen about doing reaction optimization differently. And based on the way I learned when I came into GSK and the way that I had done reaction optimization in grad school is, maybe in grad school I didn't have access to these 96 well high throughput plates with 1 ml vials, but I would typically be looking at the literature precedent when I was trying to optimize a transformation. What's the closest precedent to what I'm trying to do, the most related transformation? What are other papers that might suggest other things that might be helpful to me when optimizing this reaction? And of course, I'd use my own chemical intuition based on my experience and other permutations, trying to let serendipity play a role in reaction development as well. So typically we'll run these 96 well plates at GSK and get a diverse array of reaction outcomes at the end. And this is just a kind of red and green spot fire plot. You can think of it as a plate map of the rows and columns here in the plate, where the large green dots correspond to our hits. That's good reactivity, maybe high yield or selectivity for a chemical transformation. And then the red dots are, maybe represent that your starting material got chewed up and you didn't get any product out. So what we hope to see out of these high throughput experiments is, of course, a mix of results. It would be great if everything worked, but then you could argue maybe you wouldn't know where to go next. So we'll pick out, the top hits from these plates, and then focus in on those to design either a new reaction plate or new, single or other types of experiments to further our reaction optimization efforts. So this kind of iterative optimization practice continues, and we follow this path until we get to a point where we're happy. But we're also ignoring a lot of kind of mediocre or negative data in this process. And I was interested in doing this a little bit differently. So inspired by some of the work from Professor Matt Sigmund at the University of Utah, and I know Professor Natalie Fay has been working on this as well for some time. I was looking at, how can we quantify the diversity of chemical reagents that we're putting into our reaction plates? So I'm calling these, parameterized permutations. And I thought, if we can do that, maybe we can look at the data more holistically, the successes and the failures. So not, only using our chemical intuition and the literature resources. Let's not forget about, how those are serving us well. But really try to expand the chemical space that we're working with in these plates by quantifying that diversity and looking at these successes and failures to then understand the trends in the plates. So using perhaps predictive modeling to understand the steric and electronic features of the reagents that are impacting reactivity, and thereby, maybe predicting what to do next. So this could perhaps enhance our mechanistic knowledge of the transformation that we're trying to optimize early on in our reaction development process. And I think that could be really powerful because we often don't have much mechanistic information early on about the factors that are governing reactivity. And ultimately, that leads to new plate designs that I hope that can help us do fewer experiments in the long run. So my colleague Jen and I developed a couple of resources for our chemistry community at GSK to use here. And we focused in on, on a first pass phosphine ligands because, the sterics and electronic properties of phosphine ligands really dictate their reactivity in like palladium catalyzed cross-coupling reactions, which is a really, workhorse class of reactions in our industry. So on the left, we created this hierarchical model or kind of ligand family tree. And while it's kind of small here, you can see that there are different colors for the different families of ligands. And these are all grouped by steric and electronic similarity. So we built this based on principal component analysis of all of the steric and electronic features that we computed for our phosphine ligands using DFT or quantum mechanical calculations. And for those of you who are not familiar with PCA, although I'm sure a lot of you are, it's essentially, doing a statistical mashup of the different steric and electronic features that we feed in order to create this hierarchical model. So the way that chemists use this is in a couple different ways. One, you could look at the different areas on the family tree and select different ligands from each part of the tree for your first experiment. Or if you have something that works really well already and maybe you want to find a cheaper or more available ligand, then you can really zoom in on the family that your best performing ligand is in and look for cheaper options that are sterically and electronically similar to those that are already for the ligand that's performing well. The other option we have for chemists is this Spotfire dashboard. And unfortunately, I can't show you a live image, so this is just a static one. But we created this so that chemists could really interact with the different steric and electronic features that we've created to organize it, into a ligand map so they can design their experiments. And who doesn't love a 3D plot, right? It just looks, I think, a little bit cooler than the 2D family tree. So this was based on the same three principal components that we used for that hierarchical model. But here we're just showing them in 3D space, where the crosses are, our custom selection of sterically and electronically diverse ligands that are readily available commercially. But then the yellow to green to blue kind of color scheme shows the values for a given feature, which can be chosen from this drop-down menu. So here we have a steric feature, the cone angle that's selected, and the blue values here are basically showing the ligands with the largest cone angles, and the yellow ones are the smallest cone angles, so the smallest steric profile. And people can basically change the different feature they're looking at here. So if they want to go for a really electron-rich or electron-poor ligand, they can use that drop-down menu to see how the blue, green, and yellow color scheme changes. They can filter the scatter plot by different classes of ligands. So if they only want to look at, tri-aryl or bi-aryl ligands, they can do that. They can also drill down and see for specific ligands that they select, what are the different features that Jen and I have computed here. So again, this really helps them design those maybe second pass or third pass experiments where they really need to tune a particular feature of their ligand based on some data that they've gotten and what's successful for them so far. So now I'm going to share two quick examples of how these resources have been used at GSK. One is aiding in high throughput design, and the other is understanding high throughput reaction outcomes. So first, this is an example from our discovery chemistry group, and they were looking at this Suzuki trifluoroethylation reaction, where they needed to employ an Oompa-Loong strategy to promote the desired reactivity. So they used to have, the aryl iodide here and the boronic acid ester over here, but that wasn't working out too well for them. So they decided to switch the coupling handles. What they did was, when they designed this initial plate, they selected ligands from the literature and from their own intuition to add some variety. But they really wanted to get at a ligand not just for this one substrate, this one transformation, but a platform that would be able to do trifluoroethylation on a variety of different coupling partners. So if you look at this list of ligands, a lot of them look pretty similar to one another. There's a lot from the literature probably saying that Buchwald ligands work well. There are some bidentate ligands with really wide bite angles, probably promoting reductive elimination in this case. And in their case, when they were trying to develop this platform, only CFOS really turned on and provided good reactivity for them. So since they wanted to do trifluoroethylation on a wider array of substrates, they spoke to me and to Jen about what they could do to diversify their ligand space as they were developing this platform. So what we did was we looked at our hierarchical model, and I've just highlighted in the black rectangles here the different ligands that we chose that are all on this side of the slide. And you can see that these are from a variety of different areas of that ligand family tree. So this is kind of exploring that diversity space of the ligands that we had in-house. And of course, we chose CFOS again, just to confirm their original hit. So of course, CFOS worked, but the cool thing about this effort was that with this new design that explored the steric and electronic diversity space, they identified 3 new ligands that really turned on and provided acceptable reactivity for this platform. And this is cool, on a number of fronts. One, they get more options and can access a variety of different substrates and products from this. But 2, if any of these processes, these reactions were to come into the process chemistry space, and we needed to act really quickly in the early phase, this is really useful having options for other ligands like APHOS or like this diterbutyl neopentylphosphine that might be cheaper and more available than CPHOS is. And so process chemistry can act quickly to scale up that transformation reliably. So the second example I'd like to share is gaining some insight into reaction outcomes. And it deals with this muoroborylation, where my colleagues were trying to convert this aryl bromide into an aryl boron bond using palladium catalysis here at a high temperature. And unfortunately, when they were doing some high throughput experimentation to optimize this transformation, they observed this byproduct. So either a debrominated or a deborylated byproduct in this case. And what they found was that the identity of the ligand really influenced the amount of byproduct that was formed. So here you can see the big green dots. Those are the most successful reactions with the desired product. And then the yellow and red dots, maybe the small dots, either represent, low conversion, no conversion, or some conversion to this byproduct. So they used a variety of different ligands in this transformation. And Jen and I were wondering, in a retrospective sense in this case, what impact ligand features had, if any, on the amount of byproduct that they observed. So to figure this out, we teamed up with Professor Matt Sigman and his postdoc at the time, Toby Gensch, who is now a professor at the Technical University in Berlin. And we conducted some multivariate linear regression modeling on the reaction outcomes. We looked at both selectivity ratios for the product and byproduct and just yields for the product. And in this particular case, the best model that we got ended up looking at the yields. So we have predicted and measured experimental results on these axes, and how we create these models, not to get too into it, but we use a training and a test set split of 70-30. So these black dots are the training set. This is basically what the linear regression model is built on. And then we hold back these green dots, but then add them to the model later. So once we have this equation, then the green dots are added in and we see where they fall on the line. And these statistical parameters here give us an assessment of goodness of fit. So this was the best one that we came up with. And we were looking for a model that was also kind of interpretable, so we could understand mechanistically like what was going on with byproduct formation and how the ligand influenced it. So the different features that fell out of this model were the minimum electrostatic potential of the phosphine, which is indicative of sigma donation potential, LUMO energy, measure of pi accepting ability of the ligand, and then a buried volume, which is of course a measure of steric profile. And in this effort, we also looked at univariate correlations to help us understand what was going on in this multivariate model with different features here. So we just did reaction outcome versus single phosphine feature correlations. And this is one of the most interesting ones that we found, where there appears to be this cutoff around 65% buried volume, where the ligands below 65%, maybe like medium or smaller sized ligands, provided somewhat good to very good reactivity. And then the larger phosphines, like the Buchwald phosphines, really provided poor reactivity in terms of desired product yield. So we reasoned, this could be due to ligand speciation. These larger phosphines often lead to monodentate monoligated palladium species. And then the smaller phosphines, excuse my palladium geometry here, this could be cis or trans, of course, which has implications for reductive elimination. But these smaller medium phosphines could lead to bis-ligated palladium, which could impact product distribution. So at this point, we looked at the two optimal ligands that our colleagues found from their screening efforts. And they're these kind of mixed alkyl aryl phosphines that you wouldn't initially think of or expect to be successful in this type of reaction. but they were. So, why were these successful? Well, from the multivariate and univariate correlations that we found, we reasoned that these ligands are kind of the Goldilocks of electron density. Perhaps they have just enough electron density to act as a sigma donor, also a pi acceptor, not too electron rich, not too electron poor, to facilitate different steps in this catalytic cycle. And then their moderate steric profile could enable reductive elimination to occur from a bis-ligated palladium complex like the one drawn here. Again, this is just a hypothesis at this point. We would need more experimental information to determine whether this was the case, but give some food for thought to the chemists who are optimizing this transformation to see how they can really favor product over byproduct formation. And then for the large ligands, we thought, well, maybe if there's a little bit of water in the reaction, and we had a monoligated palladium species, so cut this extra phosphine off here, then water could attack the complex and lead to this palladium hydride, which could then undergo CH reductive elimination to form that byproduct, but get us back to the active palladium 0 species, so not quite killing the reaction. Although this reaction was parked at the time, we asked ourselves, again in a retrospective sense, how would the modeling efforts that we did have been helpful to this team at the time when they were doing the optimization? So one thing that we thought they could have done is determine the role of water in the reaction based on these modeling results and hypotheses. So looking at, is the decomposition to the byproduct or formation to the byproduct just thermally mediated because this reaction is at 90 degrees, or does it require the presence of palladium? And then design some experiments to basically mitigate that byproduct formation. Another thing they could do is optimizing ligand stoichiometry. They did, in fact, confirm that a 2-to-1 monodentate ligand to palladium ratio was optimal in this case, but we'd probably need more kinetic information as well about the nature of the turnover limiting step in this case in order to determine, is this model really, and are our hypotheses resulting from these models really correct? That's all I have there, and I hope I've convinced you in this short talk that by thinking in 3D and starting to quantify the diversity of reagents that we're putting into our high throughput plates, we can now start to model our reaction outcomes to understand trends and look at the data holistically. And get more mechanistic information early on. But also this quantification can help us efficiently explore chemical space and uncover really new modes of reactivity or new ligands that we wouldn't have thought of just with our own chemical intuition. So with that, I will stop and maybe take one question before I head out to my training.
Thanks a lot for that talk. It was very interesting. Do we have any questions? I can't see anyone jumping in. I just had one question, which was I feel that a lot of students still probably use that sort of more intuition and iterative approach. Would you have any advice for how to start to move towards something that's a bit more informed by some of the sort of methods you were talking about?
Yeah, that's a great question. And I think it's a difficult problem to solve because when I think of myself, like in graduate school, which was not, super long ago, I don't think I really had the resources available to me at the time that we do now, like with these phosphine ligands. So I know Matt and Natalie have been working a lot in this space. There are others jumping in as well. And so we're getting, I think, in the literature, a much more diverse and holistic view of the types of chemical reagents that we use. I guess I would suggest, like, if you can look at different like Mayer reactivity parameters look at more like linear free energy correlations in your data, like pKa of something that you're using in your reaction versus reaction performance or rate, then that might lead you to think about optimization like a little bit differently. And that's kind of like the simplest form of linear regression, you know, just plotting one thing versus the other. That's something that I found really powerful during my graduate school time, even when I didn't have access to all of these different features. So.
Okay, great.
All right, I gotta go. Thank you so much, Amanda. Thank you, everyone.
Bye.
Okay.